welcome Lars Henning Engelholm, who is group leader and associate professor at the Finsen Laboratory at the University of Copenhagen. Thank you for being with us here on France 24.
Thank you to be here.
Your research has focused on one of the deadliest forms of cancer, pancreatic cancer. And a year ago already, you were talking up the possibility that this antibody treatment could mean a breakthrough. What have you found since?
Yeah, so we are working with what we call antibody-drug conjugates. So it's actually a connection between very, very potent chemotherapy and an antibody. And this drug modality has made a huge difference in many cancer indications. And what we have shown in, I have to really stress that out, preclinical models for pancreatic cancer, is that we can actually cure mice with an antibody-drug conjugate. However, there is still a long way to before it can be tried in humans. There are several years, but we hope to get there, and the results really give the hope that this drug we have developed has a chance to make a difference in pancreatic cancer. Also, there's a special biology in pancreatic cancer. There's both a lot of normal cells and cancer cells connected, and the normal cells help the cancer cells, and our drug actually targets both the cancer cells and the normal cells. And that's why we think it can make a difference in this indication.
So that's with a combination of chemotherapy. Why is this breakthrough happening now? What makes this possible?
Well, antibody-drug conjugates has been around for quite some years, but the tuning of the correct chemotherapy to attach to the antibody is one thing. Then there's, of course, which receptor you try to target. This is also really key. And to be honest, I mean, let's say Big Pharma, they are very good at drug development, but they are not as good in finding perhaps the targets as we are. So many new ideas actually come from, say, universities, and then it takes a time and a lot of money actually to bring it forward, and to be interesting for, say, drug development, it's very costly. So what we can show here now is that, the drug we have now, we have also, let's say, made it in a form that it can actually be used in humans. We start with an early version that can only be used in mice, and then we mature the project. So basically, I think it's a matter of several things that combines in the right way before you get such good results.
Yeah, I want to get back to this issue of how you make breakthroughs in the lab. Before I do, just right now, for our viewers, again, pancreatic cancer, one of the most aggressive forms. It's often a death sentence for many. In fact, for 85% of those who are diagnosed with it. Is this the most promising path we've seen in recent years?
For pancreatic cancers, there's also inhibitors now in clinical trials. There's a receptor that is present, mutated receptor KRAS, that is almost present in all pancreatic cancers. And there's also hope with this, with these inhibitors. Unfortunately, cancers are very good at mutating, so almost anything you throw at them, they tend to hyper mutate around it. So it is difficult, but these KRAS inhibitors give some hope. And also there's also actually other antibody-drug conjugates that seems to have some effect. So I would say it's, we really hope this can make a difference. I would also, if you look at the how it goes with many of these drugs is that they perhaps do not cure the patients, but they could suspend the life for several years, and then you can come up with the next drug. So in many cases, it will be, I also, especially for this cancer indication, I think it will be a combination of that you can live with a cancer disease instead of dying with it. So yeah, and I think that will be the reality for many years. Of course, there will be some patients that can actually be cured by drugs like this, but there's still a long way.
So let me ask you a couple of personal questions here. How many years have you been working on this, first of all?
I started working with antibody-drug conjugates in, say, 2014. I've worked with these receptors we know about for even longer, but we started working with these antibody-drug conjugates.
Okay, so going on 12 years. And
yeah.
Would it have been possible to do this if you'd been in the private sector?
I don't think they would. I mean, I also work with another ADC that we spun out, and that is actually now in clinical trials with a company that was spun out. So I don't think the industry would ever have found these receptors. It's because we know so much about the biology, how they behave. I think that's why our research can make a difference.
Okay, so we've had in the past year these massive cuts in the United States, most notably at the National Institute for Health, where a lot of the kind of research you're talking about takes place over in the US. It's huge sums of money over there. And there's been this ripple effect around the globe. It's only been 1 year. But will there be a knock-on effect for researchers in places like Paris and Copenhagen?
I think, I mean, again, if you look at pancreatic cancer, for, let's say, investors and big pharma, they would say sometimes call it a graveyard of failed drugs. So they are not super interested in putting money into this indication. So we need public funding and private charity foundations to support work like this. Otherwise, we don't get there. And of course, a massive cut like this will delay development a lot. So I think it's a very bad idea to cut funding for, say, translational research.
And it's always a struggle, isn't it, when you're a scientist to come up with the necessary funding. What's changed in the past year or two?
So what changed a lot is that in Denmark we have a special, say, research environment. There's a lot of interest in also in translational science and spin out. So we got funding for especially this kind of say innovation. We got from a Danish foundation. It's called a Frontier Grant, which is actually meant as bringing a project from a, say, research project to something that could be a drug and could be investable. And then there's also this bio incubator where we actually spun this out in a that's the way you need to do it. Otherwise, it will just be papers, which is also important. But you need to, let's say, make it interesting for pharma and investors to further development. So you normally together with the university you spin it out in a company, and then since it is so expensive, you need that. So that has been a very big difference for us, that we got these innovation grants to, say, make our early version into a human antibody. And so that is kind of the main difference for us, and also made it possible to do the quite expensive experiments we need to do.
That model that you're describing with those grants, they make it obviously easier for you to just concentrate on working in the lab instead of going out and fundraising.
Exactly. I mean, like many other places, we are very much dependent on funding, the majority of the funding at the Finsen Laboratory is something we have to apply for. So we really spend a lot of time. So I mean, we are very happy when we get these larger grants, and that will give us some peace for to actually go in the lab and do the experiments. So yeah, we've been very grateful for that.
Lars Henning Engelholm, we wish you the best of luck with your work. Many thanks for joining us here on France 24.
Well, thank you, and a pleasure to be here.
Stay with us, there's much more to come. More news plus the day's business. And in sports, it's snowing in the Italian Dolomites and Alps just 2 days before the opening ceremonies for the Winter Olympics. The women's downhill, will it be delayed or not on Sunday? It's coming up right here on France 24.